Okay, this lecture is on chapter 8, which is RNA synthesis and processing. So there are a lot of slides in this lecture. I may split it in two parts. Okay, so in terms of what's going to be covered, we'll talk about the process of transcription. I mean, it's something we've already talked about before, but we'll get into a little bit more detail. Eukaryotic RNA polymerases and general transcription factors, right? So we'll talk specifically about what's happening in the eukaryotic cell. Also, we'll talk about regulation of transcription and talk a little bit about epigenetics. I sort of mentioned that, I think, maybe a lecture or two ago. And we will talk about it again a little bit more. You also will have a journal article that's related to epigenetics after sort of this unit, after we cover this unit and you guys have the exam. After that, you guys will read an article related to epigenetics and particularly Parkinson's disease. All right, then we'll talk about RNA processing. Okay, now, you guys know what transcription is, at least I hope. It's something we cover in Bio 111, something you cover in General Bio 1, wherever you took it. And, basically it's a process of making RNA from DNA, right? And then that mRNA that's made will then go on to be translated, okay? But a lot of what we talk about in this chapter isn't just the basic mechanism of transcription. A lot of it's about regulation, okay? And so it says here, it talks about regulation of gene expression. That's what's going to allow cells to adapt to environmental change, right? It's also what's going to allow different cell types to behave differently and to function differently inside the body, right? And so it's a super important topic to discuss when you talk about cell biology. We already said this, we know this, okay? All right, so again, just like we did in replication, you got to talk about transcription of bacteria. That's where we sort of discovered the most specifics about the process. And then we'll talk about differences between eukaryotes and prokaryotes, right? So we'll start with E. coli, start with prokaryotes, and then talk about eukaryotes from there. Okay, so mRNA was discovered first in E. coli, and also RNA polymerase was first purified from E. coli. All right, so the main enzyme for transcription is RNA polymerase, right? So it polymerizes those NTPs, right, as directed by the DNA template in the 5' to 3' direction, okay? So just like we talked about before, we're talking about DNA replication, has to be replicated 5' to 3'. When we think about a sentence with words, you have to read it from the left to the right, right? You can't read it backwards. It will not make any sense. And so it's the same idea with this. Transcription can initiate de novo, so there isn't a primer required like we saw with replication. And it's at specific sites. It's not, transcription is not the entire genome, right? So the cell doesn't transcribe the entire genome. It only transcribes those genes it's going to wish to then translate, okay? And so it says this is a major step at which regulation of transcription occurs. In terms of initiating at all, if the cell doesn't need that protein made, it's not going to initiate transcription. If it needs more of it, it's going to increase transcription of that gene. And so that's where most of the regulation in terms of how much of a protein is expressed happens, okay? And so that's kind of what I was alluding to before. All right, so in terms of the bacterial RNA polymerase, it has 5 types of subunits. So you can think of it as a really big enzyme, a really big protein, okay? The sigma subunit, and so if you guys, again, make sure you're on your review sheet and filling it in, because when you submit that assignment, you are going to take a picture of every single part of this review sheet from chapter 6, chapter 7, and chapter 8, and then it should be all filled in with your notes. Okay? All right, so the sigma subunit will be the part of that RNA polymerase that identifies the correct sites for transcription initiation, okay? And so most bacteria actually will have several different sigmas, all right? Okay, so this is showing you it here, right? Here's your E. coli RNA polymerase. This is that sigma subunit that we were talking about, and you can see the other parts of the RNA polymerase as well. Okay. Now, and again, you probably heard this word promoter before, right, in microbiology, I would imagine. And so it says promoter and initiation on your review sheet and talks about the -10 and the -35 region. So we're going to talk about that a little bit right now. So the promoter is in the DNA, right? So gene sequence to which the RNA polymerase will bind to. So if you go back and just look at this simple figure here, where this sigma is binding to, the promoter is in the DNA. Okay, so it's the region of the DNA where it's going to bind to. It is not a separate protein or anything like that. It's actually the part where it binds to. Okay, so they're about six nucleotides long, and those promoters are located at 10 and 35 base pairs upstream of the transcription start site. So that's why you see -10 and -35. They're upstream. So if the start site is here, they're upstream this way, and therefore that's where the minus is coming from. So if you think about the start site as being 0, right, and this being positive numbers, back here upstream are negative. Okay, so it says consensus sequences are the bases most frequently found in those different promoters. And we kind of had this, we talked about consensus sequence before. Okay, so they are sequences that you see most often at these two promoters. Okay, okay, and so this is showing you -10, -35. And so these are the consensus sequences that we're talking about. And again, the transcription start site we're showing as +1 here. This is upstream, this is downstream. Okay, so there have been many experiments that have shown the importance of the -10 and the -35 regions, meaning if you mutate those two regions, then transcription will not be able to initiate, okay? So genes that have promoters that differ from those consensus sequences are actually transcribed a lot less efficiently. Also, like I just said, mutations will affect promoter function. That sigma subunit actually binds to both of these regions, so both are important. Okay. So in terms of how we're going to kind of talk about the start of transcription, right? So initially that DNA is unwound, right? Just like we were talking about before DNA replication, you have to have, proteins or enzymes that come in to unwind it, okay? So the polymerase, the RNA polymerase will actually unwind the DNA to form this open promoter complex and basically allow transcription to occur, okay? Says after addition of about 10 nucleotides, that sigma subunit gets released. Okay, so the sigma subunit, right, again, which is part of the RNA polymerase in general, its function really is to recognize the promoter, right, have transcription start, but once it starts going, it's going to leave, okay? And then transcription will continue. Okay, so that's what this is showing you here. It's showing you here's this closed promoter complex. You basically need to open it, okay, unwind that DNA around that initiation site here, and then essentially transcription will start. You can kind of see that little blue line there. Okay, again, once it goes for a little bit and sort of, let's go back and say exactly 10 nucleotides. So about 10 nucleotides is not exact, right? Once it's initiated, once transcription starts, this sigma factor is released, and then transcription will continue. Okay, so again, the function of the sigma subunit of this RNA polymerase is really to identify the promoter and to initiate transcription. Okay, so again, remember this is all in E. coli in prokaryotes. Okay. So this is just zooming in a little bit more on the elongation part of it, and specifically how the beta prime subunits of that RNA polymerase are sort of holding on to that DNA. Okay, right, and so this is actually showing you that here. And in terms of what's what in this picture, right, the alpha subunit is colored green, okay, right, so the alpha subunit here is colored green. The beta is blue and the beta prime is pink, right, so blue and pink. This is what's sort of holding on to that DNA template, okay, and then also here we have the other the rest of that, the other subunit of the RNA polymerase, okay? Okay, so RNA synthesis will continue until that polymerase encounters a stop signal, okay? The most common stop signal is basically a repeat of a GC-rich sequence followed by 7A residues, okay? And so in terms of sort of, right, so that sequence, I just told you what the sequence is composed of. And because of that inverted repeat of that GC-rich sequence, it results in a segment of the RNA that can form this stem loop structure. And that basically disrupts the association of the DNA template and the RNA polymerase, and that's what actually stops the transcription. So we kind of talked about this before, right, a little bit when we were talking about the telomeres. And so in that the sequence at the end there basically causes the DNA to sort of loop around. It's kind of the same idea here, right? So this is sort of a functional way, or the sort of functional reason that the RNA can sort of disassociate there, and why that signal yields stop, right? It's because it actually creates the stem loop. Okay, so this is what this is showing you here. So again, you have your RNA being produced, right? Transcription is occurring, and then once it reaches this area here, right? You see these inverted GCs, and then the basically sequence of A's. That sequence forms this loop, okay? And then basically that causes it to dissociate. So you can think of this loop as being at the end of the RNA transcript that's being made, right? Okay. However, that's not the only way that transcription is terminated, okay? So some genes can actually be terminated by a specific termination protein called Rho, and that will actually bind the RNA itself, okay? So I just kind of told you that. I didn't give you any specifics about that, right? So again, in terms of, if I asked you about termination of transcription, right? One is due to these inverted GC repeats followed by 7A residues, forms this stem loop, and that causes it to dissociate, okay? So again, that is sort of the stop signal. The stop signal is the sequence of GC residues followed by A, causes that stem loop to form, and that RNA to dissociate. There are other examples where there's actually a specific protein, termination protein, which will bind the RNA and basically cause it to dissociate, okay? So those are two different mechanisms for termination. And again, we're still talking about bacteria here. Okay, now, if we want to talk about regulation of transcription in bacteria, and I know you did this in microbiology, okay? I know you talk about the lac operon in microbiology. We're going to review it here. But in terms of how bacteria regulate transcription, most of it operates at initiation, okay? And so there were lots of studies that basically, a long time ago that used enzymes involved in lactose metabolism to show this, okay? So those enzymes are only expressed when lactose is present, okay? So 3 enzymes involved in this: beta-galactosidase, right, which will cleave lactose into glucose and galactose, essentially break down lactose. Lactose permease will actually transport lactose into the cell. And there's transacellase here, which will inactivate these toxic compounds that'll get transported along with the lactose. So bottom line is, these are 3 enzymes that are involved in lactose metabolism, right? And so this is showing you, again, this beta-galactosidase, which will cleave lactose and break it down to galactose and glucose. Okay? Now, the genes that are encoding these enzymes are organized into something called an operon. And they're basically all expressed at the same time because they're all involved in this lactose metabolism. They really all need to be turned on at once or turned off at once, okay? And so again, they're organized in an operon. And so there's 2 loci that will control transcription, right? Something called an operator, which is O, right, adjacent to the transcription initiation site, and then I, which is actually outside of the operon, okay? So this is actually going to encode a different protein that will bind to the operator. All right, and so what this is showing you here is negative control of that lac operon, okay? And so here, right, this is showing you when lactose is present. What's going to happen? The lactose is going to bind to this repressor, right? And basically keep it inactive, okay? So if you look up here, you can see this repressor protein. So remember I mentioned I. if you notice, you can see that I is really far away from that operon, right? It's not part of the operon at all. It's another gene that encodes for the protein, and this protein is a repressor. The function of that protein is to bind to basically the transcription initiation site of this operon, right? And to keep what from binding? This is the RNA polymerase. If the RNA polymerase cannot bind to the DNA because this repressor is here, then transcription is not going to occur. And again, these are the 3 genes that basically code for those enzymes that I mentioned before. Okay. However, when lactose is present, okay, those bacterial cells want to be able to metabolize it, right, and utilize it as an energy source. It has to break it down in order, to take that glucose and then break that glucose down further to get energy, generate ATP, right? So when lactose is around, it binds to the repressor, and what does it do? It pulls it off, and therefore when the repressor is not there, RNA polymerase can bind, and basically you're going to transcribe those 3 genes involved in the metabolism of lactose, okay? Again, this is the way bacterial genes are organized in these operons, and that's the way that the bacteria regulate transcription, okay? This does not happen in mammalian cells. This is specific to E. coli. All right, so let's see where we're at in our review sheet. Again, remember to be filling this in. So we talked about transcription in bacteria. We still are. We talked about promoter initiation, and then we talked about termination, 2 different types or 2 different ways transcription can be terminated. And then what we're talking about right now is negative control of the lac operon. So I have here operator, repressor. And so remember, operator is part of the DNA here where the repressor binds. Okay? And remember here, this is another part of this, right? This sits outside, that's the repressor. Okay, so operator, repressor. This gene codes for the repressor. Okay, so negative control, meaning normally it's turned off. It is only turned on, right? Turned on meaning transcription can occur when lactose is around to basically pull that repressor and inactivate it. Pull it off of the O there. Okay. All right, of the operator. Okay, so it says mutants that don't produce the I gene. So what's this? This is the gene for the repressor, right? Express the operon even when lactose is not available, right? So again, this is just giving you a little summary of experiments that basically you can mutate different parts of this to see what happens, right? And so it says this implies that the normal I gene product is the repressor. This is what I just showed you, okay? When lactose is present in normal cells, it will bind to the repressor, right? And prevent it from binding to the operator. Okay, so again, if you have a mutation, this is saying if you have a mutation here where that repressor doesn't exist, right, then this is going to be happening all the time. And therefore, the bacterial cell has no way of regulating this. And so it's going to waste lots and lots of energy producing these enzymes when maybe it doesn't even need them. Okay. So control of transcription is mediated by the interaction of regulatory proteins with specific DNA sequences, right? So this is sort of a central principle when we're talking about regulation of gene or gene regulation or regulation of gene expression, right? I've used those terms many times, right? So think about this in terms of how this applies. When we just talk about that lac operon, which are the regulatory proteins, right? And where are the specific DNA sequences? So think about that and, make sure you understand what this is saying, okay? And so this same principle is going to apply when we talk about eukaryotes. Again, there's no operons, but there's obviously regulation of transcription, okay? And there are regulatory proteins that will bind to DNA to initiate transcription or to basically inhibit it. All right, so just to move on from here. Okay, so we're still talking about bacteria, right? So I guess I don't really have a little line on your review sheet about this, right? So this is just a little more elaboration on the sort of operon topic, right? So it says cis and some terminology. So cis-acting control elements, they will affect expression of linked genes on the same DNA molecule, right? Like the operator, okay? Other proteins can affect the expression of genes on other chromosomes, like that repressor. Remember, the repressor was, produced from a gene really far away, not part of that operon, okay? So again, cis-acting control elements will affect the expression of linked genes on the same DNA molecule, right? So that was, again, just terminology. So just a little elaboration on what we talked about. So we're really still talking about the lac operon. And again, I think I mentioned this before, it's an example of negative control. Why? Binding of the repressor will block transcription, okay? So again, just to reiterate that, negative control versus positive control, okay? So for positive control, the regulatory proteins will activate transcription. Again, remember, in negative control, like we talked about with the lac operon, the negative control of the lac operon, that repressor binds and stops transcription. In positive control, regulatory proteins will activate transcription, okay? So there is an example of positive control in E. coli. So if we talk about that lac operon, right? Presence of glucose, which is actually the preferred energy source for E. coli, will repress the expression of the lac operon, right? Even if lactose is also present. So anyway, in the past, I have shown you the details of this, showing you the positive control too, but I started leaving it out. I just want you guys to just know the negative control and just focus on that in terms of those specifics, but understand that there are also different levels of control. And then if you talk about positive control, the regulatory proteins would activate transcription. So meaning if glucose is around, it will basically, oh, I guess I didn't give an example of positive control. Anyway, that's just no negative control versus positive control. Like I said, I used to have a lot more detail here and I sort of cut that out. So negative control is what we talked about, right? Showing that repressor binding and turning off transcription. If you have, and there are other examples in E. coli and other examples of regulatory proteins. And again, I'm sure you talked about this in microbiology, right? In terms of those regulatory proteins actually activating transcription. Here, the regulatory protein in the lac operon, like we talked about, the negative control, the regulatory protein is the repressor. It binds and blocks transcription. You can also have positive control, okay? And we will leave it at that. All right. The next part of this is now going to switch to eukaryotic RNA polymerases and also transcription factors. So we'll talk about the process of transcription and initiation of transcription. Okay, so again, if you look at the next part in your review sheet, eukaryotic RNA polymerase, general transcription factors, right? And so you're talking about initiation of transcription in eukaryotes, and you'll see how it's different than in E. coli than what we just talked about. And then the rest of the lecture really talks about regulation of transcription. Okay, so I'm going to stop here and then we'll pick up this part in a second part of this.